Okay, this is the fourth installment of the Chapter 13 lecture. I apologize for letting myself get cut off in that last portion of the lecture. And I realize this is a very lengthy lecture compared to most of them, but bear with me. This will form a lot of the basis for the information that you'll need to know about natural selection and evolution for the final exam. As I was saying, this is a photograph of 4 garter snakes. They all belong to the same species, but you can see that there's a lot of individual variation here. So depending on the evolutionary pressures such as predation and substrate type and prey type, there might be some individuals within this population that are going to be more successful due to the individual variation. So variable traits in a population may be polygenic, resulting from the combined effects of several genes, which for complex patterns like what we saw in these garter snakes, that's probably most definitely the case. So there's more than one gene that's responsible for those different patterns. Or they could occasionally be determined by a single gene. Polygenic traits tend to produce phenotypes that vary more or less continuously. So of course there's a genetic component to height, and height in humans is one of those traits that varies more or less continuously. So there's not discrete height categories. We have a whole continuum of heights. However, single gene traits tend to produce only a few distinct phenotypes. As far as sources of genetic variation, we can have genetic variation that results from processes that both involve some degree of randomness. One of those would be mutations, excuse me, which would be any change in the nucleotide sequence of DNA, and sexual recombination, which I've talked about before, which is the shuffling of alleles during meiosis. For any given gene locus, which is, remember, a location on a chromosome that codes for certain traits, mutation alone has little effect on a large population in a single generation. Organisms with very short generation spans, such as bacteria, however, can evolve rapidly with mutation as the only source of genetic variation. Nowhere is this more apparent, perhaps, than in medicine. So when you get a flu shot, that flu shot is based on the best information that the researchers of the vaccine had. So they are basically making a prediction of which strains of the influenza virus are going to be worse. And sometimes they are fairly accurate in their assessments, and sometimes they're not so accurate. So there's always a chance that you can still get a strain of influenza that is not covered by the particular vaccine that you get. Okay, let's shift gears a little bit and talk about how gene pools are actually analyzed. Remember that a gene pool consists of all the alleles in a population at any one time, and it's a reservoir from which the next generation draws its alleles. Alleles in a gene pool occur in certain frequencies. Alleles can be symbolized by P for the relative frequency of the dominant allele in the population, and Q for the frequency of the recessive allele in the population. So P plus Q is going to equal 1 or 100%. Note that if we know the frequency of either allele in the gene pool, we can subtract it from 1 to calculate the frequency of the other allele. And if any of you are like me and are not so keen on the mathematics side of biology, bear with me. I'm not going to ask you to do any difficult calculations, and this is a fairly straightforward concept. So I'll try to keep it as manageable as possible for you. So genotype frequencies can be calculated from allele frequencies only if the gene pool is stable or not evolving. this is where the Hardy-Weinberg formula comes in, which is p squared plus 2pq plus q squared equals 1. And this formula can be used to calculate the frequencies of genotypes in a gene pool if you know the frequency of alleles. But remember, this only applies for a hypothetical non-evolving population. Okay, so here we have an example. We have known allele frequencies. The dominant allele here are, the frequency of that in the population is 0.8 or 80%, and the frequency of the recessive allele, which is Q, is 0.2 or 20%. So we have egg cells here reflecting those frequencies, and sperm cells here also reflecting those frequencies. So if these parental alleles combine, what do we have in the offspring? Well, we're going to have the combination big R, or p squared, which is going to be 64% of the offspring. We're going to have the PQ, or big R, little r, representing 0.32 or 32% of the population between these two different combinations here of big R, and little r, big R. And then we're going to have Q squared making up 4% of the population, and this represents the homozygous recessive allele frequency. So we end up with these genotype frequencies down here, and we calculated that simply based on the known allele frequencies in that population. Microevolution, which is evolution on a small scale, so below the species level, can change the gene pool. But how can we tell if a population is actually evolving? In order to tell if a population is evolving, it helps to know whether or not the population is not evolving and what makes up a non-evolving population. A non-evolving population is in genetic equilibrium, and this is also known as Hardy-Weinberg equilibrium. This simply means that the population's gene pool is constant over time. From a genetic perspective, evolution can be defined as a generation-to-generation change in a population's frequencies of alleles, which is sometimes called microevolution. So the evolution that we all think about on a daily basis or a casual basis would be macroevolution. That is, species actually differentiating from one another and evolving into new species. But evolution also happens at the microevolution scale, and it's more observable within everyday life. We all know that generations change over time and that generations evolve. The simplest definition of evolution is simply change over time. Okay, sorry about that, my computer actually died. As I was saying, there are two types of evolution that we really study in biological courses, and one is macroevolution, which is how species actually come to be and the diversity that we see, and the other is microevolution. Microevolution is probably easier to study just because you can focus in on an individual species and look at changes that occur over a few generations. Okay, so we have just covered how evolution can be evident in changes in a population's genes from one generation to the next, which is the definition of microevolution. But what about mechanisms? But what mechanisms cause those changes in the gene pool in the first place? Well, there are four primary drivers. One is genetic drift, another is gene flow, mutations, which we have talked a little bit about already, and natural selection, which is really what we have focused on for most of this lecture. So these are the big drivers of changes within gene pools, and thus can be thought of as drivers of evolution. Genetic drift is a change in the gene pool of a small population simply due to chance. Sorry, I'm mispronouncing a lot of things today. And two forms of genetic drift are the bottleneck effect and the founder effect. The bottleneck effect occurs when there's a drastic reduction in population size, and this causes a decrease in genetic variation. An example of this is the modern-day cheetah. Researchers have been able to look at their genome and determined with pretty good accuracy that there was a period of time in this animal's evolutionary past in which they underwent a genetic bottleneck event. So they used to be a lot more numerous. There was an event that caused local extirpations, which are smaller scale extinction events. And what happened is we had a small surviving population that ended up forming the basis for all modern day cheetahs that exist today. So the original population had a lot more genetic variation. You can think of these marbles, these different colored marbles, as various forms of genetic variation. The bottleneck effect, as it sounds, can be thought of as what would happen if you were to shake these various colored marbles that represent various different alleles through a narrow bottleneck. Not all of the alleles are going to be represented in that surviving population. Cheetahs appear to have experienced at least two genetic bottlenecks during the last ice age about 10,000 years ago and during the 1800s when farmers hunted the animals to near extinction. With so little variability, cheetahs today have a reduced capacity to adapt to environmental challenges. When I was an undergrad at Oregon State, I went to Namibia in southern Africa to work with the Cheetah Conservation Fund on captive cheetah care and research involving prey and wild cheetahs. So what I learned there is that cheetahs today have very little variation, and one of the legacies of this variation, of this very little variation in their genes, is the fact that male cheetahs have reduced sperm viability. If you examine the sperm cells of a cheetah under a microscope, you're going to see that they do not look very functional. And this is a direct result of the genetic bottlenecks that they have gone through, and the fact that they are not very robust in this particular trait is evidence of that bottleneck. The founder effect is another example of genetic drift, and it can result from a small group of organisms, which necessarily have limited variation, forming a colony that's isolated from the original population. This is likely how the Galapagos Islands were colonized. So those animals from the mainland came to the Galapagos Islands, and they necessarily had less genetic variation than the whole population on the mainland. So they essentially formed the basis for new populations on the islands, but because there was less variability inherent in those few founding members, the evolutionary path that the new populations took was limited, and as a result their current forms and functions are a reflection of those limitations. Okay, I'm going to go ahead and pause here, and hopefully I can finish up with the last segment of this lecture in the next recording.